Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Welcome to today's guest lecture, hosted by the Department of Chemistry, Faculty of Science and Technology, Universitas Erlangga. My name is Salsa, and I'm honored to be your master of ceremony for this event. Before we begin, I would like to extend a warm welcome to our esteemed guest speaker, Professor Dr. Muhammad Shahid. Professor Dr. Muhammad Shahid is a tenured professor at the Department of Chemistry, Faculty of Science, University of Agriculture, Faisalabad, Pakistan. His research focused on green chemistry of nanoparticles, biogenic materials, medicinal biochemistry, and application of biomaterials for environmental and biomedical purposes. He has supervised more than 13 PhD graduates and over 200 postgraduate students, and has led numerous national research. Professor Shahid has received multiple prestigious awards, including the Pakistan Academy of Sciences Gold in Biological Sciences, and has been recognized as one of the productive scientists of Pakistan. He has delivered invited talks in 10 countries and actively contributes to scientific development through conference and academic leadership. We are truly honored to have him with us today to share his insights and expertise with us. Today's lecture will cover a topic of great relevance and important to our field, biorecovery of lead motifs. We hope this session will provide valuable knowledge and inspirations for all of you. To officially begin, we would like to invite Dr. Alfa Agustia Widati, SSE MSE, the head of the Department of Chemistry at Universitas Erlangga, to deliver the welcoming speech. Please join me in welcoming Dr. Alfa.
Okay, thank you so much, Caca. Wabarakatuh. Bismillahirrahmanirrahim. Good afternoon, everyone. Welcome to our webinar this afternoon. We are really truly grateful to see our participants. I see only from Erlangga University, but also from University of Agriculture. Thank you so much for your attendance. And then today's webinar has been organized within the goal to providing valuable insights and learning about the bioassays for discovery of lead moieties. We hope this session will not only broaden our knowledge, but also inspire meaningful to also meaningful conversation and practical takeaways that possible that we can apply in our respective fields. Before we begin, it's my pleasure to introduce and warmly for our speakers, Professor Dr. Muhammad Shahid. Thank you so much. And then we are honored to Professor Nani Siti Amina. She facilitated for our webinar today. Thank you so much, Professor. Okay, and we believe that the knowledge and perspective our speakers bring will be valuable for us. So for the participant, we hope we encourage our participant to stay engaged. And then if there is if you want to say share thoughts or submit any question, you can just our chat box and our committee will be help you. Okay. And then thank you so much once again for Professor our participant from University for Agriculture. Okay, thank you so much.
Thank you, Dr. Alfa, for the warm welcome. Next, we'll have a brief documentation session and the presentation certificate of appreciation to our guest speaker. Invite Dr. Alfa to kindly proceed with the presentation.
Assalamualaikum. So, may I share the slides? now
Oh, okay.
to start.
Okay. Wait a minute. Salsa, have you shared oh, okay. Okay. Professor
Okay.
Muhammad Shahid is the
Okay.
certificate for us.
thank thank you. Thank you. Thank you much.
Yeah,
This is
yeah,
a
yeah.
great honor
Okay.
for me.
Okay. Thank you so much.
Thank you. Thank you.
Okay.
Thank you.
Salsa, you can stop it.
Yeah. We we kindly ask participants to turn on their cameras for a group photo. To the committee, please proceed.
Okay. One, two, one, two, three, one, two,
Thank you for your participation in the documentation. Now, with an introduction to our today's guest speaker, Professor Dr. Muhammad Shahid, it is my pleasure to introduce our presenter for today. Professor Dr. Muhammad Shahid is highly respected biochemist and tenure professor at the University of Agriculture, Faisalabad. He is widely recognized for his extensive contributions to biochemical sciences, supported by a strong record of scientific publication, international visibility, and involvement in major research initiatives. His expertise spans molecular biochemistry, analytical techniques, and the development of bioassay-based research tools, positioning him as one of the leading figures in his field. Beyond his research accomplishment, Professor Shahid is deeply committed to academic mentorship and scientific community engagement. He has guided numerous postgraduate and undergraduate students, contributed to international conferences, delivered invited talk abroad, and played important roles in academic and administrative leadership. His dedicated and to innovation, scholarship, and scientific development continues to make a meaningful impact on both national and international research. With his extensive experience in biochemistry, today's presentation promises to be both engaging and valuable for all of us. Now, without further ado, please join me, coming Professor Shahid, to begin his presentation.
Okay. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. It is my great honor to just deliver this speaker at University of Erlangen. First of all, I want to appreciate Professor Alpha Costa for inviting me for this talk and the University of Erlangen. It's just a beautiful campus, so fantastic. I really appreciated by Jamal. Whenever we sit together, he always appreciates your campus and campus life. So this is one of the my alma mater and my university that's visited by Professor Nanik and Professor uh, Alfenda and also the class, Dr. Class. So it's 120 years old. Then it's a few matrices of my university, how it stand at international level. So in the field of especially agriculture and forestry is among the top university of the world and also here in Pakistan. This come are the few pictures of my city. This famous for this known as the Manchester of Pakistan because it's a textile city. Many fantastic industries are here, and also it is also called as a science city because it contains many universities. Also, the research in atomic energy setups, uh, research institute like NAYAB, NIPG, and many others. Then it's a beautiful past of Nanik and Akhlas and Professor Alfenda. They spend good time over here with my student during the workshop time. They received from the airport and then they spend a good time over here in our university. So it's just like when half days they spend with my family and they write highly appreciate and enjoy their good life over here. And many times I receive the good messages from Professor Nanik about the altogether the setup of my home. These are the two mentor, Professor Dr. Amir Jamil on the left. He is currently dean, and uh, Professor Nawaz is former vice chancellor university of another university in Pakistan. These are the six student. I'm just look quite smart and younger in it's just a back almost 22 years back picture when I just joined the university as a lecturer. And the left, right, both are now they are currently they are professor in other university, and all the three and four girls they are also in a good position in different research institutes and universities. So it's six. These are the current uh, my student, especially if you just see this Jamalis over here, and almost they currently submit thesis. This is a short, simple profile of your guest speaker. Let's come to the science. What we want to say, we want to just deliver today about the biological assays, how we are able to lead them, use these biological assays towards the discovery of lead molecules. So it's just not a short story. It's just always a long travel right from your target site to the market and the post-market surveillance. So you have to go to the different steps. So I just want to ask, is a million and billion dollar game from you want to receive your idea, conceive your idea, till to develop a new drug. It's just a long journey. It's a long path to develop a new drug in the market. At the international and here in Pakistan, we have the different setup. This provide a good trial and clinical trial, and we have Medicine Healthcare Product Regulatory Authority (FDA) at US level, and in Pakistan we have the DRAP, Drug Regulatory Authority of it. So every country have the difference with the human health. Let's come to the and once again to the drug discovery is many time in older times we have the same type of the approaches. We want to use the phytochemical and natural product this mitigate the disease or the any health problem. So with the passage of time what they do, they modify the things, and then ultimately they leads towards the new discoveries. What we do normally in chemistry, in pharmacology, in biochemistry, and different other sciences, what we just develop the extract, perform a battery of biological assays, and then we decide whether we have the biological activity or target site. If it's okay, then we go towards purification and then establish the lead molecule and go towards the drug discovery. If it's not happening, so what we do is rightly discard and reject the extract and go ahead towards the new science. For this, we are always We have the very diversified sources in our country also, from Himalayas to the seashore of Arabic Sea. So we have very diversified geographical setup, and every geographical setup have the different medicinal biodiversity over there. So we use always interested to medicinal plant because it's always a very good source of food and vegetables. So it's already well recognized by the WHO. Almost still now, 70 to 80% people are rely on such kind of products, right from the plant sources, right from the natural products. Why so? Because they have the fantastic type of the biodiversity. In case of fruits, in case of veggies, cereals, beverages, particularly in summer and in winter, other green tea setup. And the task, if we just face this thing because of the diversity, we are always facing that some kind of the variability. Why? Because of the availability occurrence is always change, and their biological functionality, also the polarity and reactivity, and how much the lead molecule, your bioactive green compound are available in what concentration. Then we have another thing. Whenever we just go towards the extraction, we have the series of problem. It always what kind of the solvent we want to use, how many cycle we want to use, and what is the sample to solvent ratio. And then we have the particle size of our samples, time and to conduct this extraction, and also the chemical nature of your compound, whether it's a labile, it's a heat tolerance, whatsoever. There's a different type of problems whenever we are focused towards the extraction, because the extraction is the first step towards the discovery of the lead compound from the natural product. So we have the many conventional method look like maceration and decoction is a common, so I'm not going detail. Similarly, the oxalate, and then we have the steam distillation and the superheated steam distillation. So there are a variety of conventional method, extraction method. Then go towards the emerging technology like ultrasound assisted. Similarly, the microwave assisted. This quietly we in our lab we are commonly using such kind of thing. Then also we have the setup in different department of supercritical, and also one of my PhD student also have the experimentation on enzyme assisted because of my background of the biochemistry. Let's another thing is one of very emerging latest thing that's called the plant milking technology. What we do, just plant control the plant growth, and directly it's look like a milking. We rightly from the exudates we extract the uh, different biological compound from the plant source. So this is called the plant milking technology. So once a short pause, a fantastic dinner at Kanara. It's a canal that's rightly passed from that, and we at that height, and it's a beautiful setup for the dinner with the Professor Nanik, Akhlas, and Alfinda and Jamal. They all together during the conference. So it's a short pause. Let's come once again to the drug discovery. Microbial genome is also because it's easy to play, easy to culture, is multiplication speed is good, so genome size is also shorter. So it's also involved in the discovery of metabolites and also the lead compounds. So we have initially, if we just see it's a penicillin, there's streptomycin, erythromycin. So altogether, the many of Nobel laureates, they work together to this develop success story about the drug discovery from the microbes. Then we have in current time, we have the whole genome sequencing. What it do, just this a wonderful, powerful technique this provide how we are able to dig into genome, and then we target the genes that's gene of interest, and go towards the different variability, and we are able to identify that. Then actually behind this genome and gene story is we want to use different bioinformatics. If we have the genome size, then we have the different bioinformatics tool to hit the uh, targeted, mine the targeted side of gene that we are able to exploit for drug build. Similarly, we have another good science. This of the if we have the plenty of microorganism in samples, so we use the metagenomics, and this shorten the story uh, for the microorganism, and we are able to in shorter time and we have the different setup for this metagenomics to conduct research on this one. Then we have this different bioinformatics tool to use the metagenomic data into this big data into the by using, so we are able to reach anywhere to the concrete information. Sequence analysis by using blast and interproc scans, and ultimately if we just see it's a shorter story, we have the microorganism just culture, get metabolites, perform different biological assay, and ultimately you are able to get the drug. Same story the once another beautiful way. Short pause for them after the microbial science. Few students, not all, particularly they are directly involved in organizing this event. Majority of my student, that group, the student from my research group, they are present over there, along with the Professor Nanik, Alfind, and also Akhlas. You just see right behind me that Jamal is standing over there. So once again, thank you much to go ahead. This come to the new approaches in drug discovery and how we are able to use the different biological assay. So we use combinatorial molecular modeling and also the biotechnological and especially the recombinant DNA technology. So what we do in combinatorial chemistry, we have the hit, target hit therapeutic, then lead towards discovery. Then we just perform different optimal lead optimization, and then we decided this one is one of the candidate drug. And once we have the candidate drug, then we go towards the other sets of biological assays. So it's look like hitting the things. Maybe we are always think it's a molecular target, select lead molecule, and then perform the validity steps. Whenever we are targeting any lead molecules, we are always also see the site, how we are able to target. Maybe the enzyme that lead molecule opted, some kind of receptor, it may be rightly bind to the nucleic acid or any block, any ion channels or the transporter. So all these are the target site for our lead molecule. Then we perform the different pharmacological testing. It may be directly on animal or on different in vitro assays. So it's altogether it's a preclinical setup. Then also short testing about the toxicological. If we haven't the toxicological setup, uh, and it may lead towards more toxic compound maybe. So we are always interested, always perform different type of toxicological, as I say. For that we also go towards the different level like acute, subacute, chronic, or if they directly target any organ, maybe target the reproductive system or any uh, liver system or whatever we want to target. So it is always easy and to go ahead towards the preclinical trial. So it's Hippocrates saying, first do not harm. It is always good remedy for the time to do nothing. So if you are providing help for the other and don't harm, this is one of the fantastic remedy. Then the similar, we have the saying from the Charka Samhita, the medicine is always right, but the most important thing is if it's rightly do the three things: physical, mental, and spiritual well-being. And this also the currently the WHO also have the focus on three things: physical, mental, and your well-being, how together. And if it's not possible any drug without the adverse effect. It may be the shorter or the longer adverse effect. So we need to just minimize the adverse effect and maximize the benefit side of the drug. So all things are poison, paralysis, they say, but the right dose is the most important thing to treat the disease. This come back, how many main steps we have to target for drug discovery and development. The first thing is we have to target, design biological assay, then optimize, then pre-clinic, and then clinic. Why we always perform? There's a main side of my talk. Why we perform? We want to quantify or qualify any compound, any lead compound. So it always we appreciate if it's your biological assay or your assay is rapid, reliable, and robust, and you are able to reproduce your results. Why we need a biological assay? Because we want to just check the therapeutic potential and want to just say whether these biological assay they are handy to conduct in lab experimentation to shorter the time. So typically we want to just target our lead compound and mitigate the all other debris material. Whenever we talk just biological assay, it involves some kind of screening to qualify any substance on living system, directly interact with the your cells or the microbes or any chemical compound. So whenever we perform biological assay, we have to just see three things. The one is stimulus, then is there's normally our target site. There's our compound from medicinal plant, from microbes, whatsoever. Then we that compound apply on subject, animal tissue, cell lines, whatsoever the chemical, and ultimately we just what we say record the response, what happened, how the stimuli or the compound they target the subject, and what response we are receiving. Then we conduct this biological assay on whole animals, like in vivo's assays, also on different organs, and also on the lower side, like look like bacteria, fungus, and different subcellular structures. We have the different bioassays, rightly from in silico. I just already provide you a good idea about the uh, combinatorial chemistry and all the things. Then we perform those assays on non-biological assays. Then we perform biochemical assays. Then a series of in vitro assays. It may be on direct cell or subcellular structure. Then ex vivo on tissue based. Then is on the basis of their. If we want to study the interaction studies, then we go towards the NMR based. Then also in vivo. Then on animal models, then human trial, volunteer trial, and finally the clinical trial, and then we market drug. So right from this in silico, we have to reach to the clinical trial and then to the market the drug. Uh, Initially, we always, because this is very hard to perform assay for thousand and million compound, then perform the primary assay, secondary, preclinical, and clinical. In vitro screening, we have the direct hit with the target side, and using the big data mines, and ultimately we are able to optimize our drug. And then we predict their SAR, activity relationship. So the virtual screening provide us save time, money, and efforts. Altogether, it's saving the time, money, and efforts. Then we perform the different biological uh, primary assays, where we are going in lab different assays. So we have to go towards the primary assays. Then from in primary assay, we have a long list of biological assays in our lab and also going ahead in different collaborative work. Then particularly, very particularly, uh, in primary assay, we are able to predict the potential of your compound, general nature, whether the your biological assays they are able to tolerate, is a biased or unbiased, and most important is a high throughput screening. Then, if we want to reproduce your results, whether your primary biological assay is okay to support your data, and then go ahead towards its fastness, whether it's just a cost effective. You conduct your primary assay in your lab or in your university. If it's not possible, then it's just useless to say this primary assays are good. Then we have to also see the compatibility with the DMS and other solvents. Always, whenever we are just performing the primary assay, we are trying to have a high hit rate. Normally it's just a very we have, because at the primary assays we have the good number of compound, but we are always try to get the false positive results, always, or the false negative. So this is the primary thing whenever we are going to conduct these preliminary screening basic assays. We have to use them. This is one of the assay that normally conducted in our lab by using the bovine serum albumin that and denaturation assay. One of the participants, still she is over here. She's a postdoctoral student at in China now. Dr. Hina Fatima. This is the data of her article. Go ahead towards the secondary biological assay that normally we conduct on animal-based studies, toxicology assessment, ADME, behavioral studies, and also the preclinical. Altogether, this is a arena of biological assay that we normally conducted at the secondary level. Why we during primary or the secondary assay, the most important thing is you have to standardize the your compound with your standard, and we normally ask is always is a reference standard. Reference standard is used for for example, if you are conducting the antimicrobial, you have to use some kind of ciprofloxacin or anti-antibiotic. And if you are conducting of any anti-inflammatory, you have to use diclofenac sodium, such kind of compound you are used to. Without use of standard, you are not able to claim anything about, and you are not sure about your data. Then without reproducibility, if your assays are not in good enough and you're not able to receive your data repeatable, then it's useless. And the most success story behind your assay is dose dependency. As you increase your concentration of your compound, this biological assay also provide a good sight about your. So without reproducing and dose dependency, it is not good science, but it's maybe the good magic. Okay, short pause. Once again, it's a big turban. Normally in our area, uh, the highly honored people, they wear on their head this type of turban. And also I'll show you the turban at the end of this talk. So altogether, they are one of our official office. We have this turban, and this one is our clock tower. It is late night, almost a midnight time, when we visit this tower. Almost is some 125 years old tower, and it's a very famous in our city, Faisalabad, Pakistan. It's just called Clock Tower. Let's come once again, go back to the science after the short pause. Vinblastine is just a novel anti-cancer drug that was just rightly extracted from the Sadabahar. It's a plant that's normally used for development of this drug. And now this drug is chemical synthesized. It's one of the success stories. Let's come back once again. Different biological in vitro assays. It conducted normally in short lab uh, directly we use some kind of chemical or biological material to perform these assays. So it this is a short list of these biological assays. Look like antioxidant, and then we cell base, DNA, maybe, or maybe protein, RNA, or immunological. And similarly, we have this toxicity. I just show you a short blink of these biological assays at the end. Then we rightly go towards the in vivo screening by using different animal models, maybe rat, maybe rabbit, or whatever we have the access story. And accordingly, it is always your lead or target side. What whatsoever your compound you have to target that organs, animal toxicity, and then we perform different induce the disease to the animal model, uh, and also the animal models are available. That's always we are tailored already. Then we perform preclinical and clinical trials. So it's just one of the picture of my group. We conduct anti-inflammatory assay on paw. We conduct this in lab, and then con- develop the slides and perform the microscopy, and this is the beautiful results of my lab student. Always once again come to the assays. It should always be high throughput. So you are in one time, in one run, you are able to at least run 100 or 200, 300, 400 biological assays of samples in one run. So we are always using a 96, or it may be the 384 valve plate, uh, directly on using this ELISA plate or microplate reader. And shortly we are able to screen by using this high throughput biological screening technique. So look like this one. It says this one is 96 pen one plate and this 380 plates. So you in one run you can screen a huge number of compound by using such kind of plates. In a shorter way, we have this plate, and we perform the assays, and then we add up the substrate and incubate, and ultimately we run this by using this microplate. We have luckily uh, Bired and also the Biotech US plate reader in our lab from the back 10, 20 years, and it's a fantastically working word. Then these are the different setup for biological assays, directly or indirectly we do this. It's a short pause once again. It's a short. Uh, or whenever we are visiting a historical place that's called the Hiran Minar, and there is a wedding couple over there, and a class by the him to provide the turban. This one is actually class request to just have a turban. That's once again. So one of the for the departure of all the my friend guests. Then we have an cleaning in drug discovery. What we are able to do that by using NMR. So the most important, what we are doing using this NMR normally for the structure of ligand binding and dynamicity, and also we want to just get the profile of any compound, we use this NMR. This nuclear magnetic resonance system is beautifully settled for the soft tech, and we they got the Nobel Prize on directly, many Nobel Prize on NMR, but directly on uh, for the development molecule. If you want have a short molecule like drug, and you want to interact with these two, So their NMR is also supportive for this. Where different type of online maybe the LCMS and directly isolate and then split your compound for different biological assay, and also the structural elucidation by the NMR, IR, or whatever you want to use the facility of this topic. And on the other side, you just I skip this one. It's just quite common. Go towards cell line studies. In most commonly used is MTTSA. Also, we have used the different cell lines and ultimate different cell lines we use uh, to elaborate your lead compound is providing a good information or is maybe the toxic. This come to another science that's called the multi-drug resistance is quite common in drug, and that's lead towards the development new generation of antibiotics. It's just I just grabbed this picture from the Nature Face article, and almost they reported almost 60% of antibiotic drugs are not working well. Why? Because in Pakistan and in all the developing country, the pharmacological companies they are providing the backdoor money to the doctor, and doctor prescribe that whatever they want to say. So it's just the main thing behind. So the overuse or the misuse of cause of the multi-drug. Not only multi-drug, but also the currently we are facing the in case of Salmonella typhi, we are using the extensive drug resistance, and no one drug is using usable or target the Salmonella typhi. So it's another report of Cambridge. Almost the pharmacological different pharmacology pharmaceutical companies, they directly hit almost 126 billion to get the more than. So it's just a short slogan of WHO. If we have no action today, we are not able to cure the tomorrow. Why is happening so? It's easy to hit the few microorganisms, and if they become the resistance, it's hard to target. Behind this resistant mechanism, what the microorganism they do, there are many mechanism for the resistance, but the one of common is biofilm. What they do, they just perform this uh, rightly develop the EPS, extra polymeric substances, and then they communicate and telephonic system that's called the quorum sensing. This is the life cycle of biofilm. Once the microorganism they stick to surface, and in 4 5 steps they develop a good colony on the surface. In our lab, we perform this tube assay for this biofilm, and also we have the very good facility to use this microplate reader method. So in one spell we are able to use 96 or 384 compound in one spell for this biological assays, and that is optimized one of my former student, and we publish many articles in this field. Then we also have the same facility by using the scanning microscopy and also the phase contrast, or whatever you perform this biofilm inhibition assay in your lab and perform document those by using phase contrast microscopy, SEM, or atomic force microscopy, whatever the facilities. But this is one of the picture how we use these different bacteriocin against the biofilm. The same different compounds of different nature. we harvest against the biofilament, document by using the phase contrast microscopy. This is a whole film, then it's a standard, and we have this compound, different compound from the different natures. And this is one of the mechanisms that was already published in Journal of Nano Material. Not only towards because I'm working in a agriculture university, so not only for always for the pharmacology and chemistry and pharmaceutics department of farming, we are also supporting the agriculture side. So it's one of the beautiful story that we conducted experiment on the mucilage from the citrus. We in our lab we have also have the different biological assays for crop sciences, from their leaves, from wheat sample, from sunflower, from whatever the sample we get from the agriculture of departments. We conduct those experiments. Look like proteases, protein content, then peroxidase, superoxide dismutase, catalase, total phenolic compounds, also and different antioxidant elements. So once again, come to another short study that was conducted in our lab. We extracted the uh, glucanes. There's a polymer from the mushroom, and then those mushrooms we conduct bind with the nanomaterial and publish a good article, number of series. She's Dr. Hira. Then we go towards another biological assay we are normally conducting in our lab, how we are able to control the drug release, drug discovery towards drug release, how we are able to target the drug to the active site. Because it's a very common, many times drugs are liberated in the stomach, and different stomach enzymes, they degrade your compound. So target drug delivery is main target towards drug discovery. So in our lab, we have a cloning system, in vitro assays. We have the dissolution bath, and it's quite common thing that we use for these studies. Uh, she's Dr. Asma. She completed PhD, and currently she's working as assistant professor in some other university. She developed some nanomedicine binding with a mushroom, and a huge short story she conducted, a very success story from her thesis we receive, and ultimately we publish in a good number of articles in different series. And I always request to Jamal to perform this essay. Uh, this is my one of the elder son is Abdullah with the Dr. Mazhar. Uh, he's the guy behind the optimization of this assay in our lab. So this is called the hemolytic assay, or it's also called the cytotoxicity. We use this RBCs and uh, use our compound and want to see the pattern of their attack to the erythrocytes, and ultimately we read on this uh, microplate reader. Then we have another short and beautiful short assay that is called the thrombolytic assay for the CVD compound, cardiovascular. If we want to hit the compound against the CVD, cardiovascular diseases, we perform this very primary initial assay by using this simple in vitro thrombolytic activity. Then we have this short setup for a brine shrimp assay for the toxicity, especially. We have the brines, eggs, and then hatch them and ultimately perform this biological assays. Look, so it's screen for the toxicity, especially. This is the story that I start in my early career when I was just a lecturer. And this is a routine biological assay in many lab, not here in Pakistan but also worldwide. And we use this uh, different and gram positive and gram negative bacteria and we use the synthetic compound, the extracted compound from the natural sources, medicinal plant, and ultimately we use this diffusion method or the minimum imagery consideration by the microdilution method. We have a very good setup and routinely we are using, but we are now replacing this one antimicrobial assay by the biofilm inhibition, because that's a very fast and it takes longer time to perform this antimicrobial assay, and wastage of nutrient microbial mediate is also another
Be
thing.
careful, be careful, be careful. I'm done.
Okay, so this is a few findings of our lab. This one quickly. Uh, then we have also performed on different these biological assay on modified polyurethane modified cloths, and we received good results and ultimately we published in good previously good reported journals. Not only this kind but also the natural dyes also, because natural dyes are also very always good, but we have to just screen them by using the hemolytic initially and then towards the advanced assays, and want to see these natural dyes or such kind of compounds are good, or it may be harmful for the uh, so one of the group at another university in my city, they provide the sample for such kind of assays. Then it's just a marvelously good assay and it's quite handy to perform. And if you perform this essay, you are easily be published anywhere in good results and in any good reputed journal. So we perform this assay that's called the DNA damage assay or DNA damage protection assay on plasmid BR322, or also because this plasmid comparatively costly, so we perform cow thymus DNA, this DNA damage assay is on using the 1% agarose gel uh, along with the ladder. And if your compound is toxic, it provide results like this one. If it's compact like this, that's that means that your compound is not toxic. If it's toxic, it's rightly damaged your DNA, and you are get the smear in the on the gel. Then another beautiful assay by using this DNA, by cow thymus DNA, whatever the DNA you have, you directly hit your DNA, your drug molecule with the DNA by using simple UV sorption spectrometry, or if you are want to just go ahead, you may use the thermal denaturation assays. But the most commonly we are using, because we want to just see how our drug molecule they interact with the DNA compound. It may be just slightly bind with the intercalating, or they directly bind with the just groove. So is this type of interaction is normally used studies for the anti-cancer drugs. We want to just stop the uh, recycling of the DNA molecule in the cancer cells. So this type of studies normally we use to conduct for the anti-cancer drugs. Also for the other, but most importantly uh, for the anti-cancer drugs. Another high throughput assay that was conducted about the toxicity is using the LMCPA. We just dip of this LMCPA onion in the sample, and place for few normally it's a 24-hour or 48-hour. Then from the tips of these roots, we can conduct the microscopy. This is a in vitro so beautiful short essay that provide a good information about the how your compound they interact with the DNA molecules. And if it's toxic, it may uh, stop the cell that stop the cell, or it may develop the micronuclei, or it may stop the separation of the spindle of the chromosome. So a fantastic information you are able to get by the nuclear aberration of the roots, tip roots, cells of the onion. This is a this essay is really available in Pakistan, and I'm sure this your university or in your country. That's called the AIMS test or AIM assay. That was actually developed by the Bruce Aim. He was uh, in back almost 4 5 decade. He was working in Berkeley University. When we are when we want to just develop this essay, I just send an email to Bruce Aim. I want please kindly gift your Salmonella typhi strains. So then he rightly asked that they provide the patent rights to some company, and then we purchase those patent those mutant strain from the directly from the company. And we are lucky we are able to use this biological assay in routine use for carcinogenicity and mutagenicity biological assays. This is actually the shortcut of that AIMS assay. We firstly purchased from the company, and then the recipe of this essay was uh, reverse by using the reverse engineering, by using different research article. We uh, developed the recipe of this assay because it's a very costly if you just again and again request us chemicals from the company. It's quite costly, and then we just using the reverse engineering, we optimize the assay in our lab from almost more than 10 years past, and we are routinely using these assays. Comparatively costly, but their information is also important because it provides information about whether your compound is carcinogenic, mutagenic, or it's not mutagenic. So how many steps are involved in this assay? I just elaborate over here. Sorry. So when we just get the results, we are always use one sterility plate, and then we perform one background and one positive. It always a strong mutagenic compound, maybe the sodium azide or whatever we want to use. And this is the same like positive control. If it's your compound convert this violet color into yellow color, then they are providing information your compound is highly toxic. It is mutagenic, it's carcinogenic. This is a beautiful screening method that's used in our lab by using this Bruce James strains. Currently, there are 5 strains are normally used for this assay, but in our lab we have the 2 strains, Salmonella typhi TA98 and TA100. This is a toxic mining story that was conducted by Dr. Zunaira Subhani. We perform different biological assay, how we are able to bind the toxins. It's just because a big dilemma in poultry. So different mycotoxins, ochratoxin, aflatoxin, they create problem for the poultry. So it's this beautiful assay that was conducted by Dr. Zunaira Subhani and is in our lab. And then we published this article, this short story in our university. That's a success story that published in our university. 101, 101 innovative idea was published in innovative catalog in our university. So we published one innovative idea in this catalog. Short pause. This is if you just see this one picture is comparatively a bit earlier, and as this beautiful place, this is called historic and beautiful place that is called the Hiren Minar, along with my friends for this Jamal and all, and this one that's at a bit late, and if you just see this same building with a beautiful lighting system. It's a so good trip with a friend friend and Professor Dr. Nanik, Alfinda, and Akhlas, along with the other friends. So we are going towards the end story of my talk. I just want to share the few ongoing project of my student, what they are doing in our lab, and then wind up this talk. This is Arslan Rasool. He submitted his thesis. He was using gum and developing this nanobio composite for the wound healing. This is a so beautiful study that was conducted, and ultimately this submitted for the thesis. Very proud of degree to this Arslan. Then we have this Jamal that they are also conducted a very good part of his research at your university at Yelanga. So what we are doing, we are developing the polyherbal product for against the sinusites. This is a very intensive. I'm so lucky to have this Jamal in my group. He's a so lucky boy. They spend good time and always support our lab and receive a very good admiration and appreciation from the Nanik group and from the Indonesia. Then we have this another guy, this Adil. He's currently working and going to submit thesis in the next few months. He's currently working almost wind up the story of the oleo resins. He separate the oleo and also the resins. Use the oleo for the lead compound and a delivery system. She's Aisha also submitted the thesis. She's working on carbohydrate based nanobio composite. I developed different modified different nanobio composite for the drug uh, delivery, also for the binding of different nanoparticles. And we have another very hardworking student. This she's Faiza Kamal, also submit thesis. She isolate the pectin and also the lignin and develop the nano by using different biological assays, and then go towards the different characterization technique uh, in from the fiber technology, from the food technology. And conduct a very intensive study, very hardworking and very complex study she conducted in our lab and submitted the thesis for final evaluation and award of degree. Um, Another story, that's how we use different medicinal plant and ultimately use in vitro assays and in vivo assay, and that's in a battery of assays. Just imagine how many assays are conducted by this man. He is Dr. Riaz. Currently, he he completed PhD in my group. Also spent time in University of Glasgow. He is here, and currently he's associate professor in another university in my country in the field of biochemistry and allied health sciences. So, going towards conclusion of today's talk, if you want to isolate any compound, any lead compound, you have to use the primary, secondary, and different biological assays. And if you want to conduct the biological assay, you also have to just see the compatibility of your compound with the biological assay, whether your compound is on a safer side or on a safer side, or on a cell line studies and system, this quorum sensing system in our lab, and want to just secret. If I got a chance, I want to just essays. This because of my stupid expertise of in the field of biological essays. When admiration from Google, I just upload my papers and all the way myself and in my university. This is current profile of we are on the basis of entrepreneurship. We, from the different university, different corner of my my country, Kamal, they are always helping hand in my lab from these essays, and we are lucky whenever we have the even the economy or remain to purchase the chemical and all. Essays. Not only Jamal, but many other students always they pass out. They are always sporting, and they are ultimately they are get the expertise of these biological essay in our lab. Sorry. So the game that was start from the just 6 students is grown up to this number. It's all the postgraduate and the PhD student of my group. It's not class. It's my group. Like few years. My family people and I'm most welcome to Professor Alpha Acosta and the other my colleague from Erlanga University to most welcome to my university to just see my family and invite the people who appreciate sport our lab. I want to just acknowledge everyone who's supporting our lab and especially the University of Erlanga for the University of Erlanga, they support my student, and I always happy to visit them and they invite them to just join our university for this beautiful campus. And my ultimate goal is you always compete with the others for the good science with a positive, very positive mind. And if you just not able to compete with them, try to collaborate with them. And if you want to collaborate, you have to go ahead and touch the skies where you're not able to uh, alone you are not able to compete them. So it's another thing that routinely we are conducting such kind of workshop and conferences in our university, and it is one of the opportunity for many people. And every years we conduct this workshop, training workshop for the young students and young faculty about the biological essays, how we are able to support them and invite the different scientists from the world, different corner of the world for to support this workshop as an invited speaker. So the take home message is always support the other if you want to success. And if you push pull them, you're not able to success. Be positive, support them in science and arts. Go ahead. And I'm really thankful for University of Erlangen to inviting me as a guest speaker for this. I, if I support and provide something, I'm always more than happy to just support you all together. And this is all about my stupid talk. Thank you much for joining, and happy to just respond to your questions.
Yeah, the participants. If you have a question, you can raise your hand and
Sorry, can I ask a question, Tata? Miss Tata, can I ask a question to Dr. Shahid, if I'm allowed?
yeah,
Please.
sure.
Okay, thank you. By the way, thank you, Dr. Shahid, for enlightening us on a precious knowledge about bioactivity testing. Sorry, I cannot open my camera due to the signal issue. So I have several questions to you, actually. First is it mandatory to perform acute toxicity before we perform in vivo study on to discover new drugs? And the second one is, by any chance, like you already speak that the series to discover drug is very long, like starting from the in silico, and then we proceed to in cell line studies, then animals, and then human and clinical trials. By any chance, can we just jump or skip the certain step? So like we skip on the cell line and then from the in silico, and then we can directly perform animal study and then clinical trials. Is it possible or not? So that's my question, and thank you.
Okay. Yes, absolutely. You're right. I want to just respond the first question of your first question. It is mandatory to conduct the ecotoxicological assays, because if you want to just screen by using the different uh, primary assays and also using the secondary assays or in vivo studies, you have ultimately also parallelly, just if you just get some kind of lead compound, you also just go towards the ecotoxicology or any screening method. Any screen provide a good insight into your compound. If it's a safer side, on a safer side, then you go ahead. No need to be just because and if you have the access to look like this AIMS assay or other ecotoxicological assays, then you go ahead. uh, the response towards your second question is yes. By using the high throughput assays, we are able to shorten this one. But one drug is not just coming within night, in a few days. At least if you just see towards the COVID, it almost take the time. Even it's a very fast, and on the science and technology is at a just jumping at a very high level. You're not able to just pace the pace of the science, whatever we have today, as compared to the many decades back. So within almost 3 to 6 months time, we are able to develop this mRNA-based vaccine in case of the COVID-19. But for the drug discovery, you have to go through all the steps. So you haven't passed or just stop or just omitted any assays. If you want to develop the drug at the international level, and you haven't because then now that era is coming to the pharmacogenomics and personalized drugs, then another difficulty will be generated, because it's a very, and the geonetics, your environment is different, our environment is different, so it became difficult for but on the safer side, we are always asked, and we are always bound ourselves uh, to just go through all the possible assays, as many as possible assays, as many as in vivo, uh, preclinical and clinical trials possible. So on a safer side, it must be conducted. So otherwise, maybe after a few years, maybe it's become difficult. Maybe the company will be banned. It's maybe toxic.
Okay, then thank you for your answer. By the way, my name is Umi Zubaida, and I'm alumni of Universitas Airlangga. Previously, I was the student of Professor Nani, and I bought, this year. No. I mean, like, on March, I went to Pakistan, and I acknowledged that Pakistan was a very beautiful country with a beautiful culture also. So, yeah, thank you for your answer.
So thank you very much. I'm very happy.
Yeah.
The Jamal they all told about you, Dr. Musa and Akhlas all together. They are
Yeah,
it's a
no.
fantastic. He's
Now
he
Dr. Musa was in
Jamal is
Marquette.
always appreciating and admire. He's so impressed from the University of Ayalanga and the attitude of your administration, like Professor Alpha and Professor Nanik, Alfenda, and so impressed. He's always admire about your setup and your campus life at University of Ayalanga. I
Yeah,
appreciate
that's very kind of you, Professor. Thank you.
Is there any more question from the another participants? Yes. So because we don't have any question, first thank you very much, Professor Shahid, for the insightful presentations. Um, and to all participants for your insightful questions. We'll need to bring the Q&A session to a close. Now we are nearing the end of today's program. I'd like to extend my gratitude to the presenter for the excellent presentation and to all of you for your active participation. Before we conclude, please remember to sign the attendance list by our followed by the documentation. We kindly ask all participants to turn on their cameras for a group photo. To the committee, please. With that, today's event has come to an end. We extend our sincere gratitude to Professor Shahid for joining us and sharing his exercise. And we thank all participants for their attendance. We hope everyone has gained valuable insights from today's session. I am Salsa, your MC signing off. My apologies for any speech. Thank you and wassalamualaikum warahmatullahi wabarakatuh.
Thank you much, Professor Dr. Aska. This is so wonderful. Your hospitality as a just as a guest speaker, and I want to once again appreciate Professor Dr. Nanik Sati Amina and Professor Alpha Coster and your dean, all the administration of your university to inviting me as a speaker. I wish and all the best for your university, for your future, to get a highest height in your life, and always appreciate you. Whenever you want to just join my lab, my group, I always appreciate. So we have a very good setup for the biological essays, and not only here, from the different corner of my country, they join us, and we are also always receive the sample from the worldwide side different countries. This send sample even from the Europe and even from many other countries. Wherever we have our students are working, and we have the connectivity over there. They share the sample for their different biological acid, and I'll be very happy to establish such kind of biological assays whenever I just rightly visit your University of Erlanga. Uh, to establish such kind of essay at your university. I'm more than happy to support you all the way. This is my ultimate goal in my life, to just deliver the good science at all corner of the world. So with this, all my kind words, I want once again appreciate, admire your university, your administration for inviting me such a so wonderful talk, brilliant participants, and a very good, lively campus life of your university. Thank you much. I'm really sorry and obliged for your patience during the early few minutes. I interrupted during in my laptop. So I'm currently using my laptop for my wife. Thank you for your patience and tolerance to my for 2 hours. Thank you much. Any comments and words? This all together
Thank you.
and from
Yeah,
my
thank
side.
you,
Thank you.
Professor. Thank you, Prof. Lai. Thank you so much. It's okay. It doesn't matter. It's totally perfect for our very beginner today. We really appreciate for your offer, for your invitation for us to. We hope that our collaboration can we make a further collaboration, not only guest lecture, not only the collaboration in between Prof. Nani, Prof. Vinda, and Prof. Said, and maybe our students can explore campus from Prof. Shahid from there. Thank you so much, Prof. Shahid.
Thank you. Thank
Thank
you
you
very
so
much.
much. Thank you.
Thank you.
Okay. Salsa,
Okay.
we
Thank
close.
you. Thank
Okay,
you
thank you
much.
so much. Okay.
Okay, Allah Hafiz. Thank you much.
Thank you. Izin live ya.
Terima kasih Bu Alfa. Thank you also, Prof. Shahid.
Thank you.
Thank you.
Thank you.